All right, welcome back, everyone. I hope you enjoyed the ice cream. I'm here to introduce Professor Ron Weiss. He's a professor in the Department of Biological Engineering and in the Department of Electrical Engineering and Computer Science at MIT. In the early days of his lab, they mostly focused on prokaryotes, but in the last 5 years, a majority of the research has now shifted to mammalian synthetic biology, which I'm sure we'll hear all about. The lab focuses both on foundational research, like creating general methods to improve our ability to engineer biological systems. And then they also focus on pursuing specific health-related applications where synthetic biology provides unique capabilities. So please join me in giving a warm welcome to Professor Weiss. Thank you.
Thank you. Pleasure to be here. Thank you for the invitation. So I'll tell you a little bit about our work in mammalian synthetic biology. So I'm actually a computer scientist by training. And about a quarter century ago, as a graduate student, all of a sudden became fascinated with this notion that we might be able to program cells in the same way that we program computers. So I'll talk to you a little bit about that. I understand that there's a lot of computer background here. Is that true? Okay. And there's this rumor that they're trying to also move you a little bit towards biotech as well. I don't know if you've heard about that, but that's part of my role, I guess. And so I'm going to be talking a lot, actually, in terms of computer science kind of jargon, if you will, but really applying that to the world of biology. Okay, so I think those of you with a computer background, I think will understand some of the language. Those of you with a bio background may also some of it, but it's kind of going to be a mix and match of different things. But as a computer scientist, when I looked at this 25 years ago, I thought it'd be kind of cool to be able to program something like this, right? And so as a programmer, you say, I want to have this thing do X, and then you think about a program that will be able to exactly specify what you want that thing to do. And so this is something we're trying to do in biology as well. So come up with some kind of a precise definition of what we want the cells to be able to do. And then the next thing we do is actually convert that to some kind of a gene regulatory network, okay, that represents something that genes and proteins and so on would actually perform inside the cell. And then once we figure out that gene regulatory network, we actually encode that on a piece of DNA or RNA, and we take that DNA or RNA, put that inside the cell, and that essentially allows us to genetically program the cell. Now the cell can do things that it was not able to do before, okay? And so at least, hopefully at a high level, this sounds reasonable. And then you begin to realize that programming cells is not that easy. And so, for example, two of the things we're going to talk about this afternoon include liver development and cancer immunotherapy. And so then you realize that the cells don't make your life easy in terms of programming them. And the underlying networks, the biological networks, are really rather complex, right? So these are networks in liver development, and these are networks that are related to the immunology of cancer. And you see there's a lot that's going on there. These are just tiny little snippets of what's actually taking place in the biological world. And as a computer scientist, when I first began to give this talk to biologists, they said, no way, it's way too complicated. There's no way you can actually program cells to do what you want. And I would actually flip that around and actually say that in order to be able to program something this complex, we actually need tools that are as complex as the underlying biology, okay? And this is where synthetic biology comes in. This is where the notion of building programs that can modulate, the who, what, when, where, why, how much. These are the things that are going to allow us to overcome very complex environment where we need to be able to modulate many of these things. So I'll kind of build up and walk you through some of that as we go through this afternoon. In order to tackle the complexity of these biological networks, we've basically adapted something that's actually for the engineers here is going to seem fairly familiar, And what's essentially become the central dogma in synthetic biology is almost every system that's out there consists of sensing modules, logic processing, and actuation. Okay, so this notion that we build genetic circuits that allow now the cell to be able to sense new things outside the cell, new things inside the cell, senses in such a way that that information then is aggregated into this logic process. This is essentially our CPU, if you will, of the genetic circuits. And the CPU takes information from the outside, from the inside, and makes decisions about what to modify inside the cell. So, for example, modifying gene expression, protein activity, the metabolism of the cell, what the cell secretes, and so on. And so we can take this kind of notion and build several different kinds of modules. Okay, so these are the kinds of modules that we often build inside the cell. So inducing gene expression, multi-input logic, on-off toggle switches, oscillators, and feedback regulation. Okay, so these have kind of become the bread and butter of synthetic biology in terms of building and getting the cells to do something that they've never done before. Okay, and so today we're going to be focusing on multi-input logic. So going through a series of multi-input logic circuits that allows to program the cells to do something that they haven't been able to do before, but also something with therapeutic purposes. So I'll talk about that. And again, we convert that multi-input logic represented, hopefully some of you or many of you are familiar with this kind of notion. Okay, you remember what I showed you before? I guess this is going to be, oh, there we go. This is not going to become a chalk talk, I hope. And so convert this type of logic into a gene regulatory network. And again, need to be humble in the face of biology, right? Realize that our little applets are just tiny little control mechanisms. I guess this is, hopefully you guys can synchronize your vision to correspond to. I don't know if there's something and this is why I turned to biology, right? Much more reliable in electronics, right? Okay, and so I'm going to go through a couple of examples, and I'll show you here 3, and I'm going to go through 2 of them. And these are actual applications that we've been working on for quite some time in the lab, okay? And so we basically, again, start with programs, and we think about how these programs can actually do something useful. And the first one I'm going to talk about is where we build programs, genetic programs that we use to genetically engineer viruses that go inside the cell and then basically perform a biological computation to figure out whether that cell is a cancer cell or not. And if the answer is yes, then kill it. And if the answer is no, then just simply go away and not do anything. And so this is the program. I'm going to show you how we actually implemented this kind of program, which has an if statement. If these conditions are true, then initiate a cancer immunotherapy program. So I'll actually, we'll go through how you start from the basic elements and actually build a program that can actually do that. The other thing we're going to be talking about, projector willing, I guess, is this notion of building programmable organoids. Okay, so the notion of starting with stem cells and genetically programming them to create mini organs, which we then are using for doing things like drug development. So basically testing out drugs in these mini organs that sit in a petri dish, but ultimately actually take these organs that we grow in a petri dish and use them for transplantation. So for example, the thing that we're spending the most amount of time is a liver organoid. So if your liver fails, our hope is that we'll be able to actually generate a new liver and actually transplant that back into your body. So this is another program that we're going to be talking about. And yet another one, this one we're not going to be talking about, just give you another sense of something we're working on, which is genetically engineering cells and putting them into little beads, putting the beads into your body, and then having these cells basically monitor for abnormal conditions in your blood, for example. So whether it could be, low blood sugar levels, maybe, but maybe there are signals in your blood that you have an impending heart attack. Okay, so get the cells to be able to react to that and maybe, either alert you that this is going to happen or try to actually combat that. Okay, so this is something else that we're working on. So as you see that once you begin to think about programming your body, your imagination can just go wild. There's so many things there are things that we can imagine now, but hopefully, you guys will actually come up with even cooler things in the future, ways that we can actually, improve our health and so on. Health is one application, but synthetic biology, I'm not going to talk about other areas, but used in sustainability, agriculture, bioenergy, food production, environmental applications, so on. So there's a tremendous number of things that you can imagine that we might be able to do if we can program biological cells. Okay, and so let's actually start with some basics of the technology here, at least abstractly. Building blocks. Okay, so how do we actually build these kinds of programs? And so we start off with things that do basic logic operations. So for example, the NOT logic operation. So how can we build a NOT logic operation, an inverter, if you will, inside a living cell? Okay, so the inverter has a simple truth function. So there's an input, which we're going to call R, and then there's an output. So the output is always the opposite of the input. Okay, and we're going to right now just represent high and low signals, right? True and false, and so on, just as a way to simplify things. And so there are going to be two conditions. So we're going to have protein concentrations represent the signals, right? So if there's a low protein concentration, that's basically a low or zero or false, and that there's a high, obviously high would represent a true or a one. And so two cases, if the input is at low concentrations, then it allows the output to be produced, okay? And if the input is at high concentrations, it acts as a repressor and represses the output, and there's no output, okay? So we have a 0 to 1 and a 1 to 0 mapping. So at least when you make a PowerPoint, this is quick. You can make this PowerPoint in an hour or less than an hour. And then this took me about 3 years to do my PhD, just to give you an idea inside cells. Once you can build these, and I'll show you again later on how it's actually implemented in real biology, but as a synthetic biologist, now you have a building block, you can begin to have a lot of fun. Okay, so what if we wanted to do something like an AND gate out of these NOT gates? Okay, so we want a 2-input AND gate, A and B, and an output that is high only when A and B are high, otherwise the output is low. So how can we design an AND gate out of these NOT gates? So you can do something like this, where these bars are a line and a bar represent a NOT gate. So this is the way biologists would often describe biological regulatory networks. So you have input A, input B, and each one of them represses an intermediate gene we're calling R, and then R represses the output. Okay, so this is a network that we can build once we have the basic devices. So let's think about the operation of this, hopefully with a projector. Okay, so 4 cases, right? So if A and B are low, then A and B are low, that means R is expressed, and it represses the output. Okay, so 0, 0 results in 0. What about 0, 1? So 0, 1, so in this case, A is low, that allows R to be expressed. B is high, so R is not expressed here, but it's expressed there, so R represses the output. So 0, 1 still results in a 0. Similarly, a 1, 0 results in a 0. Okay, and the only time where the output is high is when A is high, B is high, they both repress R, there's no R in the system, then that allows the output to be expressed. Okay, and so there you go. So now, 3 years of work to make a NOT gate, and then many more years of work to make AND gates. Yes, question. What's that? Yeah, exactly. So that's a great question. So this is basically a wired OR. If you're used to electronic circuits, it's called a wired OR. And so the reason this is a wired OR is that either this or this has to be true. So our protein can be expressed here or expressed there, and it has the same effect. Okay, so that's the way the OR operation works. And so the other thing about the cell, and then I'll be glad to take your question. The other thing about the cell, so there are many things that are different, some things that are the same. One thing that's different is that the way these signals basically affect one another. So they affect one another not because we have a physical wire, rather we have kind of a virtual wire, if you will. All of these things are inside the cell. These molecules are like running around and bumping into each other. And the wires are molecular affinity, meaning when the protein, for example, bumps into a piece of DNA that it matches, it sticks to it, and it has the effect. Yeah. In this particular case, there is no feedback connection, although we can certainly put a feedback connection. So, for example, if we put the output protein, we might put a feedback element that causes it to bind over here. So we could. It's all within the cell. Everything is bumping into each other, but when things that don't care about each other bump into each other, nothing happens. I mean, they're like, all right, let's go on about our business. Okay, so the feedback regulation is something that you typically have to engineer, for example, by engineering DNA sequences to actually have an explicit affinity. So things that bump into each other that don't have high affinity essentially do nothing. They bump, collide, they go away. Things with high affinity bump, collide, stick to each other, and then they do their business. And then they create something. They repress something, or they catalyze the formation of something else. Okay. Yes.
So correct me if I'm wrong, but to make like the end product, it would require multiple different kinds of proteins, right? Like the final product would have multiple different kinds of proteins.
So here, the way we're implementing this is by introducing into the cell, we introduce 3 new pieces of DNA. 1 piece of DNA, 2nd piece of DNA, 3rd piece of DNA. I'm showing you the 4 different cases here. So we built 3 new pieces of DNA. Each piece of DNA consists of 2 important elements. One of them is what's called the promoter. This is the thing that recognizes, basically the protein recognizes, okay? So the promoter here is where the protein binds to the DNA and decides, you see here it gets stuck. And when it binds over here and sticks to it, let's say right here, then in this case it's a repressor. So a repressor represses the production of the protein that is downstream of it. So in this case, B binds the promoter B here, and then when it's bound, it prevents the production of protein R. Now these A and B could be proteins that are already present in the cell, for example. So if we want to be able to sense whether a cell has A and B proteins present in high levels, so they could be part of the cell, what's called endogenous proteins. For example, they could indicate whether the cell is a cancer cell or not, let's say. Okay, so when A and B are high, that means it's a cancer cell. So I'll actually go through that as an example. Okay, but then we're going to be making additional proteins. So R would be a synthetic protein that the cell normally doesn't make. It's a foreign protein to the cell. The output here could be foreign, could be natural. So for example, we want to kill the cell. So if A and B are high, kill the cell. So the output here could be a killer protein. That could be a synthetic protein that we make inside the cell.
So okay, I see. So basically, it's not necessarily like A and B are creating O. It's whether they're suppressing or allowing the production of O.
Exactly. Right. So it's a circuit, right? So they're actually not directly interacting with O. They're indirectly interacting with O, and that indirect interaction that's where the computation is taking place, right? It's that indirect interaction that they decide, A and B decide whether this copy of R is made, and then R is the one that actually decides whether O will be made. Okay, so that's the circuit. And once you realize this, you're like, okay, I want to build my own circuits, right? I want to get the cell, because these circuits are not just written in PowerPoint. We actually end up having circuits like this that we can put into a patient, right? And really begin to program humans, for therapeutic purposes or other purposes, other situations, right? That I mentioned before. So as a computer scientist, this actually was plenty for me to get excited about. Again, you get super excited, and then you spend 3 years of your life, 24/7 trying to make it work, and you realize, okay, it's a little bit more difficult than I initially imagined, but it is actually has become very doable. Because I'll show you some real examples. Okay, okay. So let's get into cancer classifiers. So let's take kind of this high-level abstraction, and let's actually begin to think: can we actually generate a virus? Can we genetically engineer a virus that will be able to distinguish between cancer and non-cancer cells and kill them? By the way, cancer is one of those areas. So for those of you who may be following, there's been just amazing progress in the field in terms of being able to, have therapeutic mechanisms that didn't exist a few years ago that can actually, be able to address different types of cancer, right? But as you probably also are aware, we're not done with cancer therapy. So many exciting things are going on. So, for example, genetically engineering T cells, these are immune cells, to be able to detect and kill cancer cells. Checkpoint inhibitors, meaning harness the immune system to be able to attack the tumors. So very exciting thing. Ultimately, it will be, most people think right now, it will be the way by which we harness the immune system is the way by which we're going to actually eradicate essentially all cancer. Okay. Now, and it's not going to be a situation somehow overnight. It just happens. It's gradual. So it's gradually happening right now. We are actually, right now, the field has generated some very powerful mechanisms by which we can harness T cells to kill various kinds of, typically liquid tumors. And on occasion, about 20%, 30% of the time, patients are cured. Okay, amazing what's going on. Similarly with these checkpoint inhibitors. Okay, how do we get to the point where it's 100%? Right, so 20, 30% is good if you're one of those people. Not so great for the rest. What can we do? So we have to think about what the challenges are. Okay, so for example, how do we find? So antigens are things that distinguish between ways by which we can distinguish between tumor cells and non-tumor cells. Okay, it's hard. So it's easier to do that in liquid tumors, like in the blood, but in solid tumors, much more difficult to do that on a regular basis. Tumors also don't make our lives easier. If we can target them, oftentimes they'll mutate. What are we going to do about the mutation? How do we make sure? So there's these super powerful technologies. I mentioned that, these T cells that can go and kill. The problem is they oftentimes kill the wrong thing as well. They're just that powerful. How can we ensure that there's no off-target effects and so on? And so I will argue that synthetic biology can really address many, perhaps most of these issues, okay, by having specificity is going to be one key ingredient in that. So it's not the solution by itself, but when specificity is combined with incredibly powerful medicine, that is a very potent solution. Okay, so we'll take a look at how we can get specificity. So this is what we've engineered. So we've taken a virus. It's a herpes virus. It's actually one of my favorite viruses, but it's an extremely good virus to work with. Okay, for a variety of reasons. And so we've taken this virus and we basically genetically engineered it to be able to do the following. So it infects cells. So it's going to infect healthy and tumor cells. And then it performs a computation, tumor or not tumor, which I'm going to show you how to do that. And if the answer is tumor, then it has a selective spread. It spreads in the tumor cells, okay? Does not spread in the healthy cells, and then begins to kill. So it begins to selectively kill the tumor cells, not harm the healthy cells, and it also interacts with the immune system. So it actually recruits the immune system. So come over here. This is a place where we need to act. It creates an inflammation in the tumor microenvironment, recruits the immune cells, and then also tells them, here are the antigens that represent, the bad cells, the tumor cells, okay? And through the combined effect, the immune system is then able to combine with a virus to ultimately, hopefully, eradicate the entire tumor. Okay, so that's the objective. And so, in order to be able to do that, I mentioned selectivity. Okay, so here is a schematic, simplified representation, for example, of tumor cells, tumor subtype 1, tumor subtype 2, and then healthy cells. And we want to make sure that our engineered approach can detect tumor cells, but basically leave alone the healthy cells. So how do we do that as computer scientists? We say, let's build a truth table. And the truth table is going to basically distinguish the biomarkers that represent tumor versus non-tumor cells. So in this case, biomarkers A and B should be high, and C, D, and E should be low. And if that's the case, then that represents a tumor subtype. Okay, and so theoretically, you can actually do that to any cell type in your body, not even tumor versus non-tumor. Essentially, anything in your body, there should be a representation, some kind of truth table that basically distinguishes this cell from all other cells in your body. Okay, and so then the question is, can we actually do that? Can we even do this in a quick plug and play fashion? So, for example, you come in, you get diagnosed as having a tumor. Can we take a biopsy and basically measure the biomarkers in that, and then within a few weeks generate a virus that's custom made for your tumor based on the biomarker? This is not complete fantasy. This is not complete science fiction. Right now, using the technology that we have, we can actually do that in 5 weeks, okay, in principle. So we can take a biopsy. Yes? So that truth table looks different for everyone. We hope that there's going to be some commonalities, right? So if you have, kidney or liver cancer of a particular type, we hope that there's going to be commonalities, that we'll be able to take an off-the-shelf, an existing virus already. So that's going to be true some of the time, but some of the time that's not going to be true. So sometimes we actually need to customize it for you. And the other thing is that even if we, target this, it's possible that, for example, targets 99% of the tumor. Right? So 1% remains, and that 1% is going to regrow. And so ultimately, we'd like to have a situation where even for yourself, as your tumor mutates over time, we have a quick plug and play mechanism to do that. Yeah. Yes. Yeah, sure. Okay, go ahead.
Just quickly, I just wanted to ask, what is the plan to deal with the natural response of biological T cells for the production of foreign viruses into the body?
Yeah, so yeah. Absolutely. So that's a great question. What is when you introduce a foreign body into the cell, how are your immune cells going to react? And actually, in this case, we actually want that. We want that inflammation that comes with the introduction of a foreign body. The reason we want that is we want that because the virus will go into, let's say it goes into various cells, including the tumor cells and non-tumor cells. And where we have the tumor cells, the virus will actually replicate. And then the immune system will notice that. And the immune system will actually create an inflammation there. And it's actually ultimately the inflammation that helps eradicate the entire tumor. So what happens with a tumor is just about every cancer, the way it's able to grow, it's a foreign body because it has mutations. But the way it's able to actually grow is it creates an immunosuppressive environment. It's able to actually cause the T cells to not be able to actually function properly. So we're fighting a war against the immunosuppressive capability, behavior of the tumor. We wanted to actually create an inflammation bring in the immune cells, and ultimately the immune cells are going to be the ones that kill most of the tumor. But we wanted to do it in a localized fashion.
Very interesting. So just one more.
Uh-huh.
So the hypothesis is that the virus attacks the tumor cells before the inflammation kills all the virus.
Exactly. Exactly. So there's some kind of a race condition, and there are actually mechanisms by which you can create viruses that fly under the radar for longer. And so it's it's a tunability thing. Like how long do you want the virus to fly under the radar and then recognize more and more of the tumor, as opposed to quickly create an inflammation. And we don't necessarily know the answers for that. That's something that we have to fine tune to get precisely right. But we have the capabilities to do the fine tuning. Yeah, but it's a fantastic question.
Yeah, so I just had a quick question. You mentioned that, if you could do this currently in 5 weeks, could you speak a little bit about what happens in those 5 weeks?
Yeah. So initially, we need to get the biopsy, so that that doesn't take that long. Then the first thing we do is analyze for the relevant biomarkers. So in this case, it's molecules called microRNA. So for those of you who are familiar with that, so we analyze the microRNA profile of the cells, and then we identify a unique microRNA profile, something like this. What is going to be a microRNA profile that allows us to distinguish the tumor cells from healthy cells? Okay. Once we recognize that, we have established a general genetic circuit that we can replace the microRNAs. Okay. So we have a genetic circuit that I'll show you in a few slides, where we have actually six inputs, and we basically say, let's find 6 inputs, 3 of which in microRNA inputs, 3 of which are low, 3 of which have to be high. So we're restricting ourselves to a particular type of topology, a particular type of logic function, 3 low, 3 high. We replace the microRNAs. That takes us maybe about 2 more weeks or so. Then we have a new circuit that is able to detect that particular microRNA profile, and then we need to put that inside the herpes virus, so that the circuit now regulates whether that virus replicates or not, whether it produces the inflammatory signals or not, and that takes us about 3 more weeks. So that's where we end up getting about about the 5 weeks or so. Okay. Yes.
So, weird question.
Yeah.
These synthetic proteins, can they also potentially mutate like viruses, which are also basically proteins?
Yeah. So this is biology. So mutations happen all the time. I mean, so something we might, we always need to consider is what happens if our virus mutates, for example. And that's true. But the way we try to approach, so we can't guarantee with 100% that virus will never mutate. In fact, we can guarantee with 100% that will mutate somewhere, some point. But the question it's a statistical game. So if the virus doesn't mutate that often, and the other thing we do is we have redundant mechanisms, such that let's say one mutation typically will not hurt what the virus is actually doing, then by and large, you will rarely get mutations. When they happen, rarely will they cause any harm. Most of the time, this thing will work very well. Right? Yes. Yeah.
The first question is, he also uses technology to program cells to become stem cells, and then also he also uses technology to cure AIDS.
Yeah, great questions. So with respect to stem cells, that's something that actually we work with stem cells more than anything else in our lab. I think they're just tremendously exciting, and hopefully, given time, hopefully I'll be able to talk about that and show you a little bit of what we do with programming stem cells, either to make stem cells from fibroblasts. So from skin, you can actually make your own stem cells. We didn't invent that. That was a Nobel Prize was won for that. But the research was done 2005, 2006. So that's technology. We're improving on that technology to make that production better. what we spend most of our time is taking the stem cells and actually creating, for example, a liver out of the stem cells. So it would be your genetically identical liver that we make in a petri dish. With respect to AIDS, that's a great question. We have created a circuit, genetic circuit, that can identify this specific proteins that are part of the virus, right? And then the idea is that if our circuit goes into the cells, it can actually detect whether that particular cell has HIV or not by detecting the presence or absence of these proteins that are unique to the virus. And then and we actually showed that we can actually kill the cells. Now, the reason this is actually not a cure is that while we can destroy cells that are infected with a virus, the difficulty is that it actually hides, the virus actually hides in your body in reservoirs that we don't really know where they are, and it's hard for us to access them. So right now, our technology does not allow us to easily get to every single cell in your body. I mean, that's just not, we're not capable of doing that, right? If we are able to do that, then we can identify and kill all the infected cells. So I think we need something else that we haven't figured out yet. So you can do things like modifications in, I think maybe CCR5 is a receptor that the virus uses to actually attack the immune cells. And so if you have this mutation in CCR5, then the virus no longer is able to infect your blood cells. And so I believe that stem cell therapy had to do with something where the new blood cells were actually mutants of CCR5. I don't know if people heard, there's also this other thing in China that where there was actually genetically engineered babies. Yikes. Okay, scary stuff. But what's that? So they use CRISPR to actually modify CCR5. Sounds like doesn't sound like a good idea. It's a terrible idea. Because CCR5 actually has its own good functions too. What's that? Sorry, yeah, go ahead. Yeah. Say again?
I just feel like it could be good because we could probably eradicate genetic diseases.
Yeah.
Also, could be a bad side where people feel like just the butterfly effect. Like you change something, it might have an on thing effect that didn't really happen in.
Exactly, and so you have to be extremely careful with what you do. You have to. I mean, you can't cut corners. I think what he did is obviously just that's just not the way science should work. No thought was given to this. There was no process that was put into place. So horrible. And then when you knock out CCR5, then the immune cells lack certain capabilities that expose you to other pathogens. And so that was actually, that was just dumb, scientifically speaking. And certainly we can't take any of this lightly. This is super powerful technology, and everything that we do in this regard has to be done with the appropriate experiments, the appropriate controls, the appropriate mechanisms that put safeguards in place. Okay, arguably this is technology that's more powerful than anything else we've ever had in our lifetime. And so there's tremendous amount of good in that, but also we spend a lot of time in the community as a whole spends a lot of time on certainly the safety aspects of it, but there's moral aspects, there are ethical aspects to this, and we could easily spend the entire symposium talking about those aspects as well, which are critical. And we do spend in the conferences that we have in synthetic biology, we do spend a good amount of time in those as well. And for example, in my lab, we had actually ethicists in our lab, kind of just hanging out with the graduate students and the postdocs in the experiments So that is, that is absolutely critical. Yes.
So I just wanted to ask, if as you go on, you could expand more on what pi and rho is, and perhaps if there is somewhat of a range for elder cells, and what to do when there is an edge case, say the range is 100 and 101, and then maybe there is already an infected cell that's going to come 101 later, or stuff like that between that point. How do you define distinctly which one will
Yeah.
become, infected, which one?
Yeah, fantastic question. So up until now, I've been using this notion of highs and lows, and synthetic biology has been to a large extent focused on like kind of this digital logic, right? And so, you can define, A has to be above this threshold, B has to be below this threshold. And, there are many situations in which that works. But one of the things, and I actually have a slides on that, hopefully, it depends on whether we have time or not. But what we've been working on lately is trying to move away from this kind of notion of digital logic, but actually work towards neuromorphic computing. Okay, so I assume everybody here has been exposed to some extent to neuronal networks and artificial intelligence, and so one of the things that's so powerful about that technology is the ability to build really good classifiers, right? Classifiers, as in you're exposed to a set of inputs, right, and you have some neuronal network that can determine with high level of precision what the output should be, like high, low, tumor, non-tumor. So we've actually taken this notion of digital logic to create classifiers, and then we've actually, instead of using AND and NOT gates, we've actually created neuromorphic circuits to distinguish between tumor and non-tumor, and they do better. So we have not published on that yet. I have slides on that, but your point is fantastic. So if you want to come work in my lab, we'd love to have you. But yeah, so I think that in many cases, this notion of neuromorphic computing, that's kind of this is basically taking back neuronal networks into where they belong, right? I think it's going to really change the way we do synthetic biology and program cells. Okay, right now, so we've done that for cancer. We've also done that for we're working on that towards organoids, to be able to actually create organoids in a better way too. Yeah, but yeah, thank you for that question. So yeah, and please keep them coming. You guys are asking some awesome questions. So let's talk about. Let me show you.
I know what story.
Oh, you know about the story about. Okay. Oh, Henrietta Lacks is a whole nother story. So I wanted to put it out there. So if you haven't read this book yet, you really need to read it. I mean, it's not a technology book, but it's really about society and some of the issues that we have been facing and continue to face in society, and especially as it relates to technology. So it doesn't delve into technology, but it creates links between the technology and the biotechnology and society. And so I highly recommend that you guys read this book. But so we're using the cells from Henrietta Lacks, to try to see if we can develop circuits that can distinguish between tumor and non-tumor cells. Okay, so that's the way we're using the cells, so they have been extremely helpful to science, even though it was done without consent. They have been extremely helpful for science as a whole. In fact, again, I don't want to get into all of the issues that arose from that, and the fact that her and her family got nothing out of it, and so on. Major injustice there. But I don't know the point that you want to make. Yeah. What's that? Are we giving the fan? No, we have not, and we probably should. Yes, absolutely. And so with these cells, with the HeLa cells, we've identified that they have a particular profile where this, so I mentioned this notion of microRNAs, right? So microRNA 141 is low in HeLa cells, and microRNA 21 is high in HeLa cells, okay? And so that is true in HeLa cells, and so you can build a genetic circuit, or if you wanted to build a genetic circuit, you build one with this kind of truth table, where you would say, if microRNA 141 is low and 21 is high, then express a killer protein called HBAX, otherwise do nothing. Right? So that's the circuit that we built. Okay, so it looks like this. So basically, this is the digital logic. So it basically says not microRNA 141 and not microRNA 21. Okay, and so the idea is that this would be true in HeLa cells. Okay, this is the logic. This is the genetic circuit. Okay, and so the way this genetic circuit works, I'll very briefly go over this. MicroRNA 141 represses the production of the killer protein, and microRNA 21 represses the repressor of the killer protein. Okay, so the only way that this killer protein is going to be made is if 141 is low and 21 is high. In all other conditions, it should not express the killer protein. Okay, so it's not that easy to cure cancer, as we kind of established. So this is not good enough. This was not specific enough. The performance was really not there where it should be, and so we realized that we need not 2, but we need 6 biomarkers. Okay, so specifically 141, 142, 146 have to be low. Okay, and so this is the portion of the genetic circuit that guarantees that the output is low only when this is low, and this is low, and this is low. When any one of those is high, that basically represses the output. Okay, and the other thing we need is this 21 also to be high, and so this is the circuit we did. And then 17 and 38 also have to be high. So I'm not going to go into all the details. I mentioned that at some point we're going to stop looking at all the details. But basically, if a cell has this microRNA profile, then it's a HeLa cell, almost with a 100% guarantee. Now, will it make mistakes every once in a while? Possibly, but it's going to be extremely precise. That's the idea. And so this is the circuits. The performance of the system, and it works pretty well, not perfect, works okay. So this is the production of the output in HeLa cells. This is the production of output in other cells. So, again, biology, right? So you see sometimes that cells that shouldn't be expressing a protein are expressing it at some level, right? So it's not quite perfect. And again, this was about a 3-year project. A lot of projects are 3 years. Many of them are more than that. And so, but not bad. So we can now distinguish between HeLa and non-HeLa cells, and then we can actually, in terms of the specificity in killing, the circuit actually kills the HeLa cells really quite well. And this is the data to show that. And the worst behaving cells, the HEX cells, it doesn't kill that much. Okay, so it has pretty good performance, enough to write a paper about, but not quite ready for prime time, not quite ready to be used in a clinical application. And so Jin, a former postdoc in the lab, said that he wants to redesign the circuit. So he took the circuit that I showed you, or the general notion I showed you, and he started off with these types of regulation, and then he added what's called a feed-forward loop, and another feed-forward loop, and another feed-forward loop, and a feedback loop, and another feedback loop. And this was not 3 years, this was longer. Sorry, I forgot about the other feed-forward loop. And this is what he created. Okay, don't ask me to explain to you how this works. Okay, this is an insane circuit that he made over quite a few years. for dealing for being able to create a cell classifier that would work very well. But after having done all of that work, he actually got really good results. So now he's getting over 100-fold sensitivity by creating this insane circuit. Okay, so and we pretty much understand how it works, and it has a whole bunch of a question about redundancy. So it has a whole bunch of redundancy. If certain aspects of it fail, then there are other aspects of it that are probably going to work and make sure that it's kind of fail safe. So that one we said, okay, we can begin to think about putting that into mice. And in fact, we have some preliminary results in mice showing that the virus, when we inject it into, this is a 4T1 breast cancer in mice, actually replicates, this is the kind of negative controls, and this is the classifier that works really well. It replicates just in the tumor, and it actually doesn't replicate in the rest of the body. Okay, so it's actually rather specific, and it also, in terms of survival, has significantly increased survival, and it actually is also we inject it in one place where the tumor is, but this tumor is highly metastatic, meaning that it goes into other parts of your body, and this mechanism is actually in cancer, 90% of the patients end up dying from metastasis, not from the primary tumor, but rather from the metastatic lesions. And this here is actually able to really target metastasis really quite well. It really is able to eliminate metastasis. So we're excited about that. And we think a good portion of that is because it has a high level of specificity that is able to replicate in the tumor. Then it goes into your bloodstream, travels to other parts in your body, and then when it finds the metastatic lesions, it begins to amplify locally there as well. Okay, so we have some results from that. We also have a version of this now that works not with DNA, but actually with RNA. So similar to the way we have the RNA vaccines, we now have a mechanism to actually encode a cell classifier in a piece of RNA, so that the idea is to get injected by these beads, nanoparticles, which you get injected with right now when you get a vaccine. And when you get a vaccine right now, you have the RNA, and it's going to make the spike protein, which is the antigen, to allow you to be protected in the future from SARS-CoV-2. So here the idea is that the RNA first computes, is this a tumor cell or a non-tumor cell? And then if the answer is yes, it actually amplifies itself and then creates this local inflammation with RNA. So we are working on that. We actually have experiments that we've done in mice, and now actually begin to work with non-human primates as well to see whether we can actually take that to the clinic. So we're super excited about that. Okay, let me talk about organoids, talk about what we want to be able to do with stem cells as well, okay? And so, as I mentioned, this is an area where we're probably spending, I don't know, how am I doing on time, by the way? I think I have a minute. Okay. Whoops. Okay, so we've done about a third of, so you guys have had fantastic questions. Really appreciate that. So we've done, so I'll just go to the last slide. Just, I'll go through any questions. Yeah. Oh, you have one? Okay. By the way, this is a liver organoid that we actually generated from stem cells. So as I'm going through this, yeah. No. So gene editing is a mechanism with CRISPR. It's just a mechanism to be able to modify a particular region of DNA. So you can think about that as a tool for being able to modify certain mutations that you might have in your body, or the way we actually use CRISPR quite a lot as a way to bind a protein to a specific region of DNA. But we oftentimes will use it just to bind and act as a repressor, for example. So not to cut, but rather bind, repress, or bind, activate. So if we want to repress any gene in your body, if we want to activate any gene in your body, or if we want to do that for circuits that we synthesize ourselves, we can actually use CRISPR technology to do that. But we're using some aspect of CRISPR, in various kinds of circuit technologies that we're developing. Yeah, CRISPR has been fantastic to work with. So we talked about the microRNA cell-based classifier. Wanted to talk about organoids as a way, to basically build customized organ that would be, we've been able to do that, make liver. That's basically your genetically identical liver. And we have actually put that into mice, and we're able to show that we can, restore some liver function. And we're now beginning to think about, can we get to clinical trials? If you have liver failure, can we actually, use your own stem cells to recreate your liver? And then thinking about, mentioning how to use neuromorphic computing as the new way to program biological systems. Okay, so with that, lots of people that have been responsible for the work. I think I mentioned some of them, and then the funding agencies, and then my advisor Tom Knight, that got me hooked on synthetic biology, 25 years ago, and it's been the best drug ever. I just can't get away from it. And I want to thank you for your amazing questions.